All right. Hello, everybody. Welcome to this month's meeting. Thank you all for your attendance. We have a few introductory slides before we get to our presentation. ASM Microbe, the abstract submission portal should open sometime mid-November. I don't know if the sessions yet are out, but that also should happen sometime soon. ASM policy and advocacy, which is incredibly important right now in the... Ooh. Sorry, I heard a feedback. Okay, we're all good? All right, great. So learn more about ASM's efforts to benefit all of us working microbiologists as they try and get the Congress and federal government to act on our behalf. The next month is the Eastern 54th Annual EPA ASM Clinical Microbiology Symposium. I'm not sure on how much of the spots we have open left, but I think that it is filling up all the spots. Oh, keep attending if you're not signed up yet. And keep sending it out to everybody. And then December 1st is the Infection and Immunity Forum. And then this monthly seminar series will resume in January with an industry speaker. The government has been in a shutdown since October 1st. I'm sure there's a myriad of ways that all of us have been affected by it. The shutdown risked undermining the public health by disrupting necessary services that federal agencies provide. We've been trying to get in contact with the CDC for a while about some ongoing projects. We haven't been able to hire a postdoc because of the issue with federal government. So ASM is looking for ways of learning how this has been impacted you personally. So please scan the QR code and then let your voice be heard by sending an email to your legislator, helping them to encourage and fund science. Why advocacy is important. Science really extends beyond the laboratory. There's a lot of different ways that both the intersection with politics affects how we can get and do science, and then how our results are interpreted throughout in the world. We're very knowledgeable about science. We're very good at educating people who know less about science. And so it's important that as scientists, we sort of take that foot into the public domain and try and advocate on our behalfs. This is an additional QR code you can scan if you're really interested in advocacy and working with federal and local policymakers on how microbial sciences can be represented. Very fitting that this year's or next in June ASM Microbe is in Washington, D.C., which also makes it very easy for us to go as Philadelphians. And this is the final QR code where you can share your story with ASM Microbe as a scientist. And with that, I'm happy to introduce our speaker, Medni Anapajala. She is joining us down the street from the Children's Hospital of Pennsylvania, of Philadelphia, where she's also a professor at the University of Pennsylvania School of Medicine. She did her undergraduate on the other side of the state at Carnegie Mellon University, and then moved to Columbia for a PhD in chemical engineering, and then remained at Columbia, switching to the medical campus for her postdoc. Her PhD work as a presidential fellow at Columbia was focused on metagenomic and metatranscriptomic surveillance of wastewater. And then as a postdoc, she transitioned more into applications of wastewater analysis in regards to multi-drug resistant organisms and viral surveillance. And I'm very excited to hear what you have today to just talk about your work at CHOP. Thank you.
Thank you. Great. Oh, thanks. So just a couple of slides on a circuitous path to getting here, but wastewater is a little, happening now. It's a little cooler to study wastewater, but at the time of my PhD, it was decidedly not cool to be spending my time, especially in the summer, in wastewater treatment plants across the country. But really the goal there was to understand these complex microbial communities that we actually use to treat wastewater and recover nutrients for other downstream applications. Through a few of those types of projects, we started noticing all of these antimicrobial resistance genes, clinically relevant bacteria, et cetera, all growing and to different degrees thriving in these wastewater treatment plants. So my natural next question was, what's happening to these bacteria as they enter the waste stream, and specifically coming from human hosts? And so that's when I started my postdoc looking at drug resistance and MDR bacteria, multi-drug resistant bacteria in humans, mainly through the microbiome, but also infectious isolates in the hospital system. And so the pictures are my postdoc mentor, Anne-Katrine Uleman, and my close collaborator and friend, Angela Gomez-Simmons, and just 4 key points from the work there that sort of sets the stage for what I'll present today. And the first is that by studying many patient microbiomes, especially those who are immunocompromised for different reasons, we noticed diverse colonizing genotypes of multi-drug resistant bacteria in their guts. We also realized that if you're colonized, meaning the bug is around in your gut microbiome, it's a significant predictor of subsequent infection with that same strain of bacteria. And this has been shown by not only our group, but others. On the bacteria side, we noticed that plasmids, which are these mobile elements that often harbor the antimicrobial resistance genes, we were noticing that they were being transmitted across bacterial species very routinely, especially in certain families of bacteria. And we found evidence of the same clone of an organism infecting patients widely distinct in time. So over 10 plus years, the same exact strain of bacteria or the same exact plasmid was found in patient isolates spanning that large time frame. And so these sort of 4 concepts we were ruminating on these, and I think it pretty much led to 2 major questions. One is we need a better way to capture what's the full diversity of all these multi-drug resistant organisms in hospital patients and in the hospital setting. And also, we need to identify reservoirs of these bacteria within the hospital that could explain why patients a decade apart, years apart, were suddenly getting infected with the same strain. Where are these sort of living in the meantime, if you will? And so as a wastewater engineer, I thought of wastewater, obviously, to study these things. And there's a few reasons. Traditionally, if a patient's in the hospital, they get infected with something, the clinicians and team orders a clinical sample. It goes to a clinical microlab. Robert can tell you more about that. And it gets, characterized. What was the organism? What drugs is it resistant to, et cetera. So you get these individual results for a specific patient. You get really specific info about that bug, but it's really labor intensive at the scale of, hundreds of patients, thousands of people in a neighborhood, for example. And it ignores what we found before, which was that asymptomatic carriage or colonization of these things in the gut isn't captured if you're only looking at isolates that cause infections. Now, wastewater instead is telling you more things like, these are the levels of a given pathogen on this day. How does that compare to baseline? And how can we use that information to track things like outbreaks or emergence of novel drug-resistant species over time? That has some sort of initial pros you can think of, which you can cover a big population at once. One sample is basically like collecting samples from anywhere from 100 to 500,000 people at once if you're looking at a wastewater treatment plant. So much lower cost, less labor per person that you're capturing. But there are cons, it only works if your pathogens shed in wastewater. So some people sort of instantly are like, we can test for everything in wastewater, but it needs to pass through the GI tract and enter the wastewater. So that's sort of a initial consideration, let's say. These are aggregated results for so many people, which is great and cost effective, but then how do you actually work with that data? What can you do if you see a signal? That's still a sort of open question that I'll talk about later. And there aren't really any established pipelines or standardized pipelines at all. So we're kind of flying blind a little bit here. And so a lot of my work is on establishing those methods, first of all. And then again, it's a sample getting samples from hundreds to thousands of people. The limits of detection are quite low. We need a lot of a virus to be present to detect at these scales. So there's a few sort of problems, let's say, with um, these approaches. So we do have some technical limitations. So there's different techniques we use, but they're very dilute sources. Often, if you think of a single patient in a whole hospital having an infection with something, how do you know that you can capture that if you collect all the wastewater from the hospital? And there's different techniques to try to get that. We do a lot of sequencing of the wastewater, both targeted sequencing of individual microbes in the wastewater, but also what's called shotgun metagenomics. So just everything that's there at once. And, you know, the input you get is really fragmented. Microbes are flying through these wastewater pipes. They're getting broken up. They're being lysed open. Their DNA is getting sort of floated around. A lot of stuff happens. So sequencing from these samples can be quite difficult as well. And different standardization methods are often needed. The next kind of consideration we think about are sampling scales. What level of population are you trying to capture by testing wastewater? And it's a balance between, like we talked about, getting cost-effective data on a large number of people versus what can you do with that data. So, you know, with COVID, for example, as I'll talk about later, many people tested at these huge wastewater treatment plants, and that's wonderful for public health data and tracking, but you can't find a single person who's infected with something from that type of scale. So that's something to think about. And lastly, what I'll sort of focus a lot about on today is microbes are very different from viruses and viral surveillance in the environment because they are living in the environment and they're persisting there and not just sort of passing by like a virus or viral RNA. So they're evolving to their environment, they're adapting to their environment, they're competing with each other in the wastewater and in these environmental sources. They're sharing genes through horizontal gene transfer, all of these things. So what you see at the end is not necessarily what happened upstream, unlike for a viral surveillance. So and lastly, I'm not going to really touch on this today, but sort of the future directions we're looking at is how do you actually make this type of data actionable and who's involved and who should be involved. There's also ethical consideration, both pros and cons of using wastewater instead of individual testing. I'm not going to bore you with history or anything, but this is not a new concept ever since the sort of traditional cholera story. People have been using wastewater for surveillance for different microbes. So polio in the 1940s was sort of the or the oldest modern use of wastewater testing, but then hepatitis A in the 80s, neurovirus in the 90s, actually, and then sort of perhaps at the forefront of most people's minds is SARS-CoV-2 most recently. Fortunately or unfortunately, I was in sort of New York City, the epicenter of this in the United States, when SARS-CoV-2 hit. And, you know, this is just a snapshot of the cases we were seeing at our one hospital. Huge diversity of viral strains, as most people saw in the news and through research cases. And, you know, we needed a better way. We were sequencing every single clinical isolate in our lab, but that's very cost intensive and labor intensive. How can we get at this in a better way? And so, again, we're not the only ones who thought of this by any means, but sort of had a unique experience working with both my PhD advisor and my postdoc advisor, all three of us as co-investigators on a grant. And really, we were funded by the National Institute of Drug Abuse, of all people, to study SARS-CoV-2 wastewater surveillance methods. And the reason is, you know, I'm going to talk a lot about microbial surveillance using wastewater today, but pharmaceutical surveillance, other metabolite surveillance, all of that's possible with these wastewater samples. So they sort of spearheaded this. Our specific role in this consortium of groups that was funded to look at COVID in the wastewater was to look at different sampling scales, actually. So pretty much most of the other groups were looking at these huge wastewater treatment plant levels. But as you go down in sampling scale or lower and lower population covered by a wastewater sample, there are pros and cons, as you maybe can imagine. So we sort of landed on the hospital, mainly because we were located at a hospital, but also we're interested in what's actually clinically relevant. And it's sort of a nice middle ground between getting information from a lot of people and having clear stakeholders like hospital administration or infection prevention teams that can go do something about it if you have a sort of result. But just to say, there were also dorm buildings, other types of places where this was useful. So, and this recently came out, but we sampled wastewater from two different parts of our hospital and from our own research building, actually, that we were located in. And so those are the three subsampling locations. And we were interested in this because, again, if you're trying to figure out what can a hospital do about something, if you can tell them, hey, this part of the hospital has way more SARS-CoV-2 infections than this part, they can have more tailored interventions that can happen. And so that is sort of what we found. By and large, the wastewater sampling actually anticipated. So it gave us signals before clinical testing up to 14 days. And it was a unique signal in the two different parts of the hospital. So we could tell which one had more patients with COVID at any time. Most excitingly for me as a sequencing person and bioinformatics person, on the right image, the top two panels are purely from sequencing wastewater for different SARS-CoV-2 strains, which are the different colors. And the bottom panel is our own sequencing of every clinical sample that came through the hospital. So you can tell, it's clear that each part of the hospital had some differences and distinct dynamics of the different strains, but by and large, it did recapitulate what we would expect at this hospital population level. Okay, so that was a little aside on viruses, but my title was about multi-drug resistant bacteria, because now that was sort of many people's next step is we can do this for viruses. How do we adapt this to different targets and microbes? So our first test was, can we grow them? Can we see them in the wastewater? And I think unsurprisingly, the answer was yes. You can kind of culture a microbe from anywhere, basically. These are just raw wastewater from our hospital. Yeah. And so these are, on the left, it's just all bacteria, all gut-related bacteria. We have, different levels of multi-drug resistance over there on the right, and a clear diversity of colonies. It's not just that we're picking up one or two of these resistant bugs, it's a whole community of them. And just as an aside, that's my favorite picture of a plate that I've ever taken, but it's on a fungal plate. I did let it grow for a few extra days, but it still all came from the wastewater. So, okay, so that was sort of our first test. We can grow it from the wastewater. What next? Is it just growing everywhere? We can't really tell a signal, it doesn't really mean anything. And so we wanted to see, we were sampling from the hospital, right? But also our own research building. We expected that the hospital should have, a much higher bacterial load in their wastewater. More people actually living, staying there overnight, let's say, more of a residential type setting than our research building. People are in or out. So we did find through qPCR of the wastewater that the hospital wastewater, thankfully, had a higher burden of bacteria at all sites. And you can see the green versus the blue, which are different parts of the hospital, had different overall bacterial load in the wastewater. And these are same volumes of wastewater collected at two different places, treated all the same and normalized to sort of internal factors within the wastewater. And so overall, our feeling was, okay, this is a viable way that we can try to monitor MDR bacteria moving forward. But there are these open questions, right? I alluded to this before, but is what you see what was originally there? We can grow all these things, but what does that mean for what happened in a patient? And then the next evolution of that is, what's the actual clinical relevance of seeing these things in the wastewater? I think these are two questions I'm not going to answer in this talk, but it's sort of where the field is right now, I think. And so many things are part of both of these. The microbial dynamics during the wastewater transport it's a mixed community of bacteria, and anyone who studies any mixed community of bacteria knows it's not a static thing, right? These bacteria are competing in real time. They're sharing genes through horizontal gene transfer, plasmid sharing, et cetera. But as an engineer, another piece of that is what's the impact of the built environment? You know, the different piping systems, pH, temperature, seasonality, you know, all these things. And then what are the different metabolites, nutrients, or contaminants like disinfectants or even antibiotics in the wastewater themselves? And how are those shaping the communities that we see when we surveil the wastewater? In terms of the clinical relevance, what is the relevance of finding asymptomatic carriage versus just infectious isolates like we can do with wastewater? Does that matter? Is that, you know, necessary? I would argue for different things it is necessary, and for different goals it's not. And then how do you define if a bug that you're seeing in the wastewater is purely environmental? Is it a bug that is generally a commensal gut microbe bacteria that is likely to be coming from people? Or is it a likely pathogenic strain? How do you even start teasing all those things out? Very complicated. And lastly, again, I talked about this a little bit before, but what can even be done if you see this data? And that's maybe the most open question of all. So you know, my postdoc, I was doing a lot of microbiome studies. Others who have done microbiome studies may be familiar with the general workflow where you take fecal samples, you're, you know, either culturing out bacteria from these and studying the individual bacteria from a fecal sample, or you're doing something like metagenomics where you sequence everything at once. Our sort of complement to this is collecting the wastewater samples. They have a mix of everything, right? Bacteria, RNA, DNA, viruses, bacteriophages is a new sort of interest in my lab. It does have fungal cells, some fraction of human cells, you know, a lot of things in there. And what we do is actually use different concentration methods to try to get at these different fractions. So we actually try to separate out what are the fungal species that are there, what's the whole bacterial fraction, what's the viral fraction of the wastewater. And all of that, again, goes through similar techniques as a microbiome sample of culturing out individual bacteria to sequence them and characterize them versus doing more metagenomics type sequencing where you're getting information on everything at once. And so really our goal to begin with was to characterize and quantify sort of the hospital microbiome and more importantly, the hospital resistome, let's say, of everything that's resistant to a certain class of drugs. And so we were luckily in a position to do this cool study where we were collecting wastewater, sink drain swabs, and had access to clinical isolates in our own lab, collected all from the same units of the hospital. So we were really able to see these things side by side. And all of these types of samples, we cultured out these two classes of multi-drug resistant bacteria called ESBLs and CRE. Happy to talk more about that, but I'm doing sort of a broad overview. And we included isolates captured via each of these testing modalities, along with genomes from clinical isolates that we had previously sequenced. And this was quite a huge collection. And as I mentioned before, spanning over 10 years, actually, but, you know, over 400 isolates resistant to carbapenems, you know, hundreds of isolates resistant to extended spectrum beta lactamases, and added on to hundreds of isolates from the hospital environment. So huge kind of collection there. And what we did was long read whole genome sequencing on all of these isolates. So it's kind of, you know, my favorite technique of all basically is to run long read sequencing to try to basically piece together the entire bacterial genome, but also plasmid sequences in the bacteria, which are difficult to do with other types of sequencing. And what we did was first step was who's there, you know, identifying the bacterial strains that are there, running genotyping approaches, but also identifying which actual resistance genes were causing different resistance phenotypes. And then because we had this collection of the different types of samples, we wanted to run phylogenetics and network analyses of, you know, how closely related are these? Are the things in the sinks only in the sinks? Are the things in the wastewater only in the wastewater? And lastly, we care a lot about resistance harboring plasmid sequences, as I've said a couple of times now. With the long read sequencing, you can actually close loops on the plasmids. And so we were doing a lot of work on, are the plasmids we're seeing in one collection the same as others? So is it sort of likely to be traveling through these different sample types? Do we see plasmids that exist in their entirety in different species? So is there evidence of horizontal transfer of a plasmid from one species to another? And if there are different plasmids, can we track the evolution of those plasmids over time in each collection? So again, you saw those plates. There's just a high prevalence of multi-drug resistant bacteria everywhere. I just highlighted in pink sort of the main takeaways here, but 60 to 85% of all sink drains, regardless of when we sampled them, are growing multiple colonies of these bugs, and 100% of the wastewater samples. So um, next step was, okay, they're growing something. Is it the same something? And so we looked at, this is sort of a crude Venn diagram of just at the species level, which species are overlapping versus distinct to wastewater from unit 1, wastewater from unit 2, and sink drains from unit 1, sink drains from unit 2. Those are the 4 sort of categories there. The takeaway here is, the darker the color, the more number of species that are sort of unique to that setting. There's some shared species across all of the combinations of wastewater, hospital, sink drain, but there's also a lot of species that are unique to a single site. And so this is both good and bad, I guess. If something's really unique to one site, maybe that means we can get a clearer picture of linking that to clinical patients who are in that site. Or is it a clear answer that this is really just about that location? This is just about sink drains in that part of the hospital. It doesn't really mean anything. So we had more digging to do there. So we characterized all the antibiotic resistance genes across these sites, and KPC is, the most common gene responsible for carbapenem resistance. And one allele type was present in both hospital quadrants, very prevalent across those sites. And another KPC type was, only in one quadrant really versus another one. So I think there are these two competing stories of there is this hospital-wide reservoir of these things, but also there are specific signatures in each part of the hospital that hopefully we can track down and link to clinical samples. And so that was the next step, is can we start linking these to clinical samples and across the sink and wastewater sampling sites. And so this is all of the multi-drug resistant Enterobacter cloacae complex, one of the species we look at, across all of these collections. So it's sink drains in red, wastewater samples in green, and clinical samples in blue. And the different shades of each color are just over time in each of those collections, basically. And it's a phylogenetic tree, so the closer they're clustering together, the more closely related they are. And not that you need to, digest the whole tree, but you can see clear instances like this where all the colors are really mixed together. And if you actually look at the SNP distances, the individual nucleotide differences between these isolates, in some cases they're very nearly identical across these different sampling types, indicative of either a shared reservoir that's causing these microbes to be present in all 3 collections, or direct sort of introduction from one to the other. Just another example of E. coli from the same sort of setting. And again, there are definitely clusters or nodes where it's, all sink drain isolates. So that's clearly something to do with that sink drain. But there's also, interspersed clusters like this where you do see evidence of sort of all 3 collections converging in different parts of the phylogenetic tree. Now that's at the genome level that I just showed in those 2 network plots, but I mentioned we're really interested in the plasmids. So in these types of organisms, the plasmids really run the show in terms of antimicrobial resistance. The gene is on the plasmid, and the plasmid is not necessarily tied to a specific species of organism. So especially those two species I just mentioned, Enterobacter cloaca and E. coli, they've been shown to, transfer plasmids across each other. They're closely related enough where this happens. And specifically with this type of resistance, carbapenem or beta-lactamase resistance, this happens very frequently. And so very exciting. But this is how sort of new resistance genotypes emerge in some cases by a species that wasn't really resistant to much, suddenly acquiring a plasmid that has these genes, and now it can sort of hold its own against certain antibiotics. That's not the only way that new resistance emerges, but it's one of a few different ways that can happen. And so what this is showing here is we wanted to know, we have sink drains, we have wastewater, we have clinical isolates. How closely related are these plasmids across these collections? And most importantly, if we just look at all the plasmids we have, how many of them show up in just one collection versus two collections or all three collections, basically? So which of these are most likely to be persistent across clinical sink drain wastewater? Which of them are pretty much just unique to one site? And so the two colors are those two different resistance genotypes I was talking about. So blue is KBC, which is sort of the, let's say, higher level resistance for the sake of today. And the darker blue is still multi-drug resistant, but sort of one step down, let's say. And so the two sites bar plots, sorry, I should use the pointer, but I have this mouse for now. If it says two sites, that plasmid cluster was found in either sink drains and clinical isolates, sink drains and wastewater, or wastewater and clinical isolates. And if it says three sites, it was found in all three. So for both of these types of plasmids, you can see, at the bottom here are the numbers, 700 plasmids that had KPC on them in our collection. They formed about 50 clusters. Out of those 50 clusters, I mean, you can tell from the numbers, a majority of them are found in multiple sites actually in our collection. And same thing for the CTXM, which is the extended spectrum beta lactamase. So a few key takeaways from this work, really pervasive multi-drug resistant bacteria throughout the hospital in all these different collections. The predominant species in each collection, in each part of the hospital, were different, but there was evidence of shared species, shared plasmids across all three sample types and across different parts of the hospital. And then we are proposing that probably both site-specific features of the built environment and unique features of the patient population housed in each of these locations are probably contributing to this. And so that was all work up in New York, but now I just want to touch on some of my more recent work here in Philadelphia at the Children's Hospital. And so here we again started with sort of the same setup where we have two different wastewater samplers in the main hospital at the Children's Hospital, and they cover different parts of the hospital. One, the top one is covering the emergency department and the NICU, for example. And the second sampler is covering sort of a majority of the main inpatient units, let's say, in a different part of the hospital. And so we collect wastewater 3 times a week since this past January when I sort of opened the group. And right now we have almost 1,000 of just these two phenotypes of bacteria stocked. So again, 100% of the wastewater grows these, so it's not very hard to get a lot of them, but I'll talk a little bit about the diversity in a second. We've sequenced about half of what we have so far with the long-read whole genome sequencing. So we're building this, again, robust collection where we can look at both the genomes of this species of organism, but also the plasmids located in each bacteria. So again, we're doing similar analyses here with the long-read whole genome sequencing. And so this is just a quick initial snapshot of the data. Each color is a different species. The top row is KPC harboring organisms, so that carbapenem-resistant phenotype, and the bottom is ESBL harboring organisms, so that next level down, resistance phenotype. The two panels are two different wastewater samplers, so different parts of the hospital. And not that you need to link every color to what it is, but hopefully it's, pretty obvious by sight that each sampler looks different from the other. So each part of the hospital does have this sort of unique diversity of each type of organism. And based on the resistance phenotype, there's a different profile of which species are sort of resistant to each type of antibiotic. Now, when we look at the plasmids in these organisms, it gets very fun because we do have multiple instances where the same plasmid backbone is found across different species, basically. So these are just some examples of our favorites right now. But we have different ESBLs, we have different combinations of these resistance genes co-harbored on the same plasmid and found in different species. Um, and this is sort of not only species we'd expect, but some species we really didn't see coming. So, you know, just for anyone more clinically interested, Enterobacter cloacae, Klebsiella pneumoniae, we sort of expected to see these in this collection to a large degree. But things like it's kind of buried here, Ralltella, Citrobacter, they do cause infections, but we didn't expect how often we sort of encountered them in our collection. And so our next step is really looking at the metagenomics, so everything that's in there at once. And what we gain from the sampling here is both whole microbial community profiles, everyone who's present in a single sample of wastewater. And also we can use that same metagenomic data to individually reconstruct bacterial genomes and plasmids from the metagenomes. So what's the benefit of doing that? Well, right now, the technicians are wonderful people who sit there and pick different colonies off of a plate and sequence them. So with the metagenomics and really getting it to a place where we can reconstruct the genomes from that, it just reduces all of the sort of hand picking and growing and stocking we need to do. Um, so again, I think I mentioned before that the methods are not standardized for any of this. It's actually very easy to grow a bacteria from wastewater, but it's very difficult to thoroughly sequence all of the DNA from a wastewater sample, especially with long read sequencing. So a lot of what what we've been doing, I would say, for the past 4 to 5 months is really methods development using a lot of both mock and real microbial community samples. How do we lyse open all these diverse bacteria without shredding our DNA? How do we accurately sequence everything? And in our control samples, are we getting exactly what we thought we would from each of those samples? And so far, what we've done is basically build out both the wet lab and bioinformatics pipeline to do this. And this is just an initial snapshot of the type of data we get. So these are from real sort of wastewater metagenome samples, and it's just a representation of the diversity of all different bacterial strains that we're seeing in each sample. So you can imagine it's a lot of data to digest and sort of distill down into that piece that I mentioned in the beginning, which is how is this actionable? Like, I can't just give this circle to someone in infection prevention control and tell them to do something about it. So, what we're working on now is what pieces of these are the most important and how do we translate that into something a clinician or hospital administrator can do something about. And so the way we're thinking of doing this and what we've started doing is this phase of sort of building what the signature of a piece of the hospital is in its wastewater. So we do really dense sampling of the wastewater and metagenomics at high sequencing depth for a short period of time to get this baseline, what we're calling the pan metagenome of that location. And so each wastewater sampling location has its own baseline. And we found at Columbia too that this is largely true, even though if there's an outbreak of something or an overgrowth of something, you can tell that from the metagenomic data. But beyond that, there is sort of a stable community that you find in each part of the hospital. So that lets you more easily know when something's wrong or off because you do have some sort of baseline to go off of. So we've that's what we've been working on, is building those baselines for each part of CHOPS hospital for right now. And now we're in the phase of now moving forward, what do we do and how do we sort of inform infection prevention? And so what we're doing is each week then we sequence new wastewater and map it back to its own baseline from that site. Next week, map it back. And each time we map it back, we're also building what the baseline is. So it's sort of this iterative approach where we're continuously getting a more and more complex baseline for every site, but it lets us sort of quickly get data each week instead of having to build out this really robust picture of everything that's there at a given time and just get at what's different now than it was before, and what's different to a level that we can see with just sequencing at a lower depth, basically. And so on the right is sort of our bioinformatics approach of what actually we're trying to report. And so we're, a lot of my postdoc work was in the microbiome. So we're, heavily borrowing from the microbiome field here, but diversity within a sample has just been used as the snapshot measure for many microbiome studies in the past, of just to see at a global level, has something shifted in the community? And so that's our first readout, actually, just has something significantly shifted compared to baseline at this part of the hospital. And so we use, diversity metrics, calculate what is an outlier versus the baseline. And those are the samples that we then sequence further to figure out exactly what's different, exactly what's going on there. And those are the samples that would lead us to sort of alert in the future, either infection prevention or any follow-up molecular testing of individuals. And really the vision is, surveillance with wastewater is sort of the hot topic now, but it really is beyond surveillance. We can see these things in the wastewater. We know we can see them and track them over time, but how do we use that information as a tool as sort of this next phase of where the field is going? And so we're imagining it as sort of a modular system of environmental sampling within a hospital, for example, where wastewater is sort of the biggest snapshot, collects from the whole population. It's your initial sentinel. You get those diversity metrics. Something's wrong. Something's different. Then you can follow that up with other types of environmental sampling, which we're setting up now, which is air sampling in different hospital units. We're setting up a different sink drain swabbing in different parts of the hospital. But by having wastewater sampled from different parts of the hospital, you know where to look for something downstream. And beyond that, then we can start partnering with clinical labs of, okay, now we've actually narrowed it down to a set of rooms or a unit. Now, can you tell us more about the patients here? Because we can't, there's hundreds of patients in this part of the hospital. We can't go right now to the microlab and say, someone might have an infection of something in this part of the hospital. Tell us who. So that's sort of how we're seeing it, is wastewater can be used to basically stratify what's the risk of an outbreak happening in this part of the hospital versus this part of the hospital. And beyond that, more from a bacteriology perspective and micro perspective, how do we use that info to identify these persistent reservoirs of these microbes in the hospital and use that information at a more basic science level to understand their evolution adaptation over time to their environment? And that's where sort of the more engineering piece comes in, because we've started building these wastewater biofilm model systems. A lot of my PhD work was in designing and running lab scale bioreactors to mimic what's happening in a wastewater treatment plant in a controlled lab scale. And so we're applying that approach to wastewater piping, basically, and building out these piping models so that we can test if I add more antibiotics, what happens to the resistance level within the waste stream, for example. And lastly, can we use all this information that's coming together to actually address the problem? If we know we have environmental reservoirs of something, having this sort of robust environmental sampling approach is really the only way to know the extent of your problem, where the problem is, and maybe strategies to tackle it. So one of our ideas that one of my postdocs is working on now is looking at the bacteriophage populations, for example, in the corresponding parts of the hospital and how that relates to the bacterial communities in those same locations, and starting to come up with, could we, forget about patients, can we do environmental treatments, basically using different approaches, including this. And so those are strategies to actually address the reservoirs before they become a problem. And so I want to leave a few minutes for questions. So just want to thank my young and upcoming lab that just opened about a year ago, and a lot of collaborators and funders along the way. So thank you. Should I go for it?
Yeah.
Yeah, go.
Like, use outbreak. You can air out like this one room or floor of the hospital. I mean, it's the opposite where like you kind of test that, where if you have, oh, we know that this like you can smell flush, like plumbing has a Clostridia or something, and like a C. diff.
Mhm.
Can you go one patient with C. diff?
That's a great question. We were able to do that with SARS-CoV-2. So the threshold was we needed at least 2 patients based on just like where our samplers were located to have known cases to be able to detect it in the wastewater. But for microbes, we don't know that limit of detection yet. Yeah. But that's definitely something we're interested in for sure. Yeah.
Right, thank you for the interesting talk.
Thank you.
You said went to a new hospital. How long would it take, or do you know, how long would it take to establish that baseline of, community wastewater pathogens to then use as your baseline to look for differences?
Yeah. So I started at CHOP last September, and we started sampling in January. And so far, based on our data, it seems like a 6 or 7 month period of just dense sampling to get at that. You can get a baseline. I guess it's a relative baseline, right? Because it is an ever-changing field. But I would say around the 6 month mark, you start less and less additional information is gained with each successive sequencing. Mm-hmm.
Like in New York sampling, you saw more seroplasm, CTXM plasmids,
Mhm.
but clinically you see definitely more CTXM series. So I just thought you could comment on.
Yeah, totally. I think in this case, it was more KPC specifically than CTXM 15. So that might have been part of the disconnect that we were specifically looking at CTXM 15 because at Columbia, that was sort of the main allele we were seeing clinically. But we were surprised because not only were there more plasmids with KPC, but more of the sink drains grew KPC positive isolates than ESBL isolates, regardless of the gene that caused that resistance phenotype. A few things. One is that if you look at the species that are contributing to that, that was what I was kind of mentioning of we were kind of surprised by some of the species we were seeing. So a lot of the KPC positive organisms were maybe lesser clinical relevance. So one, basically a hypothesis that we're working with right now is that there might be these sort of keystone environmental species that it's not about the clinical infections with those ones, but they're just serving as microbial reservoirs for the different genes and plasmids, and they're more responsible for shuttling them across species than for actually jumping out and causing infections. So that's what we're working with right now. I think we didn't have quite the setup there to really test that. So that's what we're hoping to do here. Mhm.
Two questions.
Yeah.
The first, since CHOP is creating a new patient tower, now you do have this access to the new environment. Do you have plans?
Yeah.
Like, you know where that wastewater is going? Like, you know,
Yeah.
So you get some sort of like, get a lot of gut anaerobes, bacteroides,
Mhm.
we also get the more kind of classic water bugs like a Comomonas and a Ascoborax.
Mhm.
So just like, what do you think is kind of, if you're a bacteria in this environment, like what do you think is going on in terms of who's living in a biofilm, who's kind of like passing through, and sort of like what are nutrients there?
That's a great question. Yes, I like most of the past year has been just calls with like facilities and plumbing teams at both shop. And so this is most of my work anyway. But we sat with sort of the blueprints of both main hospital and the new patient tower to try to identify those sampling sites. And we got clearance to actually sample for 3 months before they opened the patient tower, less, not weekly, but at some frequency, just to get at when patients start coming in, like the seeding of those pipes. And so that's exciting. To answer your second question, that's actually our main site where we're going to be asking those questions because we can see a clean pipe into biofilms forming. But in the lab, the wastewater model systems that we're building have like biofilm growth media, not media, what am I trying to say? Scaffolds. So it's a special type of bioreactor where you're testing both suspended growth of microbes and biofilm growth. It's called a mixed bed biofilm reactor. And so you can really get at who goes into the biofilm and who goes into suspended growth. But I can tell just anecdotally, it's it's very technical, and yet when you go to the wastewater collection site, it's a tube and a hole in a pipe. And so we have to sometimes, you have to like move it around if it's not in the right spot, etc. But we did this interesting test that actually Liz and Cassie, the two techs in my lab, designed and just did one day, and I was like, that was great. But they were like, well, sometimes the wastewater looks really dark and sedimenty, and sometimes it doesn't. And so what's happening there? And they thought maybe it's the tubing. And so they did a series of water tests to figure out if the density of the tubing, like where was the tubing actually sitting? Was it sinking to the bottom of the pipe, or was it really in the wastewater flow? And so they found that at one site it had been sinking, at one site it was kind of floating. So they went out and ordered the type that just floats in the middle of the waste stream, so that we know right now both sites are getting suspended growth bacteria. Now we are interested in the biofilms separately though. So for that, you can either switch to a heavier tubing. They have tubing weights where you can specifically weight the tubing to get the biofilms, or it's just a hole in a pipe. You can stick a swab in there and sort of swab the actual biofilm, which is something we're planning to do. But I think it's so hard in the real systems to really tease that out. So I think that's why the lab scale reactors are more probably designed to answer that question. Yeah.
This is a little bit hypothetical. Wonderful presentation. Thank you for this. You were growing bacteria. Whichever bacteria is in nature, it's likely to grow in your place. One of these days, you're gonna find something that you were not expecting.
Yeah.
And it's gonna be something bad, like a select agent.
Mhm.
So are you keeping an eye to that possibility that a select agent like Bordeaux Pseudomonas might show up with one of your plates? And are you ready for what comes out? That's
Yeah.
one.
Yeah.
You answer that.
Mhm.
Number two, a multi-drug resistant plasmid has been identified in the Yersinia pestis. Very bad.
Mhm.
Okay. And you might see that one day on one of your plates. So what would you do?
We have an extensive infection or institutional biosafety committee protocol that outlines exactly what we're supposed to do when we step. No, that's a great point, and that's why, when we do the selective plating, it's almost nicer because we know what we're sort of selecting for in those plates, but those non-selective media are the sort of problem. So we do everything in a biosafety cabinet. Even though usually when I was doing microbiome work, we would just grow things out on the bench, but because it's wastewater, we do everything in a biosafety cabinet. So to answer your question of what would we do, we have to notify EH&S immediately if we see something growing that should not be growing. And yeah, that's a wonderful question.
Getting at it,
Yeah.
you to throw it away.
No. But the problem is we wouldn't know until we sequenced it. And by that point, we've sequenced it. You know what I mean? Like there's no mechanism for us to know ahead of time until it's too late, or like we've already sequenced and stocked it. So so it wouldn't be thrown away, but that probably causes problems from a institutional level. But I swear they know this is a possibility. I wrote it out in the protocol. But that is a great point, and I hope and don't hope that I will have to encounter that anytime soon. Yeah. Mhm.
Where, that's and will.
Mhm.
And was also collecting wastewater in the facility. To your question about like how much of the wastewater is like actually capturing, was it, like what was like the diversity index, I guess, from the stool versus the wastewater? Did the wastewater capture most of the stool, or do you see like?
Yeah, that's a great question. And so I think sort of along the lines of what Robert was saying of like he was noticing a mix of these gut bacteria, but also environmental sort of water related bacteria. It is interesting that from the sink drain swabs, it seems very environmental microbe heavy and water microbe heavy, whereas the wastewater does more closely mimic like a human microbiome sample. I guess when you think about it, that makes sense, but sort of when people design these studies, that's not always at the forefront. I would say I don't have a great quantitative number for you, but just back at the envelope stuff, I would say like actually 80 to 90% of the wastewater samples are just gut bacteria. I think probably that has to do with the fact that we are collecting from the suspended, the aqueous phase of the wastewater that's just passing through. I think if we did in our downstream studies of like what's in the biofilms that are sitting there, I think we'd see a very different distribution. But that's a great question. In the suspended growth, it does pretty well capture a human microbiome sample. I would say in terms of diversity measures, for example, human gut, I think, maybe in the 3 to 4 range for like a Shannon diversity, if that's what we're talking about. I would expect it to be more like 4 to 4.5 in a corresponding wastewater sample. So I wasn't monitoring the chat. I don't really know how to do that while presenting, but I don't know if I did.
Hello, I'm online. I don't know if you can hear me.
Yes, we can hear
Oh,
you.
great. I was wondering how concerned should we be um, to view the hospital as a point source for downstream uh, first selection and then release of antibiotic resistant bacteria and/or antibiotic resistance plasmids. About 25 years ago at the University of South Florida with Professor Jody Harwood, I worked on a project that looked at sediments at wastewater treatment plant outfalls in marine waters to look for evidence of antibiotic resistant vibrios that are sediment associated bacteria. So I'm curious to see are the wastewaters treated prior to release from the plants, or could they actually be serving as point sources? Thank you.
That's a great question. I think it actually depends on what city or locale you're in of how extensive your wastewater treatment is. So coming from New York, almost all or a majority of the plants there use what's called biological nutrient removal, which means using a microbial community to transform nitrogen, phosphorus, and carbon from the waste and try to remove it, or at least recover it for downstream purposes. So in those types of plants, they have done studies of testing influent different parts of the wastewater treatment process and the effluent. And they have found serial reduction in sort of antibiotic resistance genes at each of those steps. So that's nice. I think in a hospital wastewater setting, it's very unclear the directionality of all this, right? I mean, I'm saying surveillance for a reason. It's because are we just capturing what's coming out of the patients? Could this in turn cause infections in patients? I think now with data sets like these that are so longitudinal, we can start asking those questions. But I think in a lot of the literature, it's just been sort of, I have clinical samples from here, I have some environmental samples from here. It's very hard to know the directionality of what's going on. And so right now, if you even think about like how would wastewater suddenly start infecting patients, right? You would think it's just in a pipe. Hopefully the patients are not then re-exposed to wastewater. But so I think how we're seeing it is more this sentinel of what's going on in the hospital. I would find it very physically hard to believe that they're causing infections at the location where you're sampling. But to your point of you were testing near the treatment plant, I'm sure that these then travel through the pipes. Different cities have different sewer system types. If you have combined sewer overflow, where your stormwater and sewer water goes in the same pipes, takes one big storm, you know what I mean? So these things are different based on where you are. And also the population density and sort of layout of the city. New York City has 14 wastewater treatment plants, Philly has 3. And the population is obviously larger in New York, but not that much larger, where Philly with 3 plants is probably quite dense in their wastewater collection. So I think all these things matter. I think there have been a few studies of looking at riverbeds near effluents of wastewater treatment plants, like you were mentioning you were doing in South Carolina. But overall, I think treatment of wastewater, whether it's microbial or through reverse osmosis UV disinfection, it has been shown to reduce the antibiotic resistance genes with serial treatment steps. So I think I would be most concerned of where is that influent coming from and how has it traveled on its way to the treatment plant. And that's why we're so interested in piping models and what's happening in the pipes before it gets to a plant.
Thanks so much. Fascinating work.
Thank you. Cool. Thank you.